Somewhere beneath the surface of a tropical reef, in the shallows where the water runs warm and the sunlight fractures into shifting columns of gold, something extraordinary is happening. A creature no longer than your hand is about to deliver one of the most powerful strikes in the natural world. Not a shark, not a barracuda, not anything with teeth or size or obvious menace. A shrimp, a mantis shrimp, and what it is about to do will shatter glass, stun prey, and confound the laws of what a small animal is supposed to be capable of. If you have never heard of the mantis shrimp before, prepare yourself. Because once you understand what this animal does, how it does it, and what it costs the body to do it at all, you will never look at a coral reef the same way again. And if you have heard of it, if you have seen the vivid photographs, the explosion of color, the dramatic descriptions online, then stay with us. Because the real story is far stranger and far more precise than any headline has managed to capture. Before we go any further, if you find yourself captivated by the hidden physics of the natural world, by animals that push biology to its absolute limit, then make sure you subscribe right now. There is more of this to come, and you do not want to miss it. Let us begin at the beginning, not with the punch, but with the animal itself. The mantis shrimp is not a shrimp in the way most people understand the word. It belongs to an ancient order of crustaceans called stomatopods, a lineage that has been evolving on this planet for somewhere in the region of 400 million years. That is older than the dinosaurs, older than the first forests, older than almost every animal group that most people would think to name. While the continents were still being arranged into recognizable shapes, while the first amphibians were just beginning to drag themselves onto land, the mantis shrimp was already here, already perfecting itself, already hunting. There are more than 500 known species spread across tropical and subtropical oceans around the world. They live mostly in burrows, in crevices in the reef, in self-excavated tunnels in soft sediment. They are solitary and intensely territorial. They will defend their home with a ferocity that seems absurd given their size. And they will attack almost anything that comes within range, including the hands of divers and the fingers of marine biologists who should have known better. They range in length from a few centimeters to the largest individuals, which can reach nearly 40 centimeters. They are armored in segments, built like a tiny submarine with articulated plating across the thorax and a tail fan that flares out like the spread of a hand. And they are, without any serious competition, the most visually remarkable animals on the reef. The colors alone are staggering. Mantis shrimp can be vivid orange with electric blue spots. They can be striped in greens and pinks and purples that seem to have been chosen deliberately, flamboyantly, as if the animal is well aware of how extraordinary it looks. Some species are comparatively subdued. Others look as though they were painted by someone who had been told that subtlety was forbidden. The coloration serves purposes both in communication and in camouflage, depending on species and context. But it is not the colors that have made the mantis shrimp famous. It is the club. More precisely, it is what the mantis shrimp does with the club, and how it does it, and what happens in the tiny sliver of a second between the beginning of the strike and the moment the prey realizes it is already dead. The mantis shrimp broadly divides into two categories based on the structure of its raptorial appendages, the specialized front limbs used for hunting. There are the spearers and the smashers. Spearers have spiny appendages lined with sharp barbs, and they use them to impale soft-bodied prey like fish and shrimp, stabbing outward with precision and speed. They lurk in burrows and ambush animals that pass overhead. Their strike is devastating in its own right, but it is the smashers that have captured the imagination of scientists and engineers and anyone who has ever had the fortune or misfortune of getting close to one. Smashers have evolved a club-shaped appendage at the end of the raptorial limb, a dense, rounded structure made of layers of material so sophisticated in its construction that materials scientists are still trying to fully understand it decades after first studying it. The smasher uses this club to attack hard-shelled prey. Snails, crabs, clams, the heavily armored animals of the reef that most predators simply cannot crack. The mantis shrimp cracks them. It does so with a strike of such extraordinary speed and force that the physics involved seem almost implausible when written out in plain language. Let us talk about that speed. The strike of the smasher mantis shrimp is one of the fastest movements recorded in any animal on the planet. The club accelerates from rest to its peak velocity in a time measured in thousandths of a second, reaching speeds of around 20 to 23 meters per second. To put that in context, the strike is faster than a bullet fired from some handguns. It is certainly faster than the blink of a human eye, which takes somewhere between 100 and 400 milliseconds. The mantis shrimp strike is over in less than 3 milliseconds. You would have to be watching through a high-speed camera running at thousands of frames per second to see it at all. But speed alone, remarkable as it is, does not fully explain what happens. When the club connects with its target, two things happen. The first is the obvious impact, the direct mechanical blow, the transfer of kinetic energy from the moving club to the shell or body of the prey. This force alone would be impressive. Studies have measured peak forces from mantis shrimp strikes in the range of hundreds to over 1,000 newtons, a force many hundreds of times greater than one would predict simply by looking at the animal. A creature that weighs just a few tens of grams should not be generating forces measured in the hundreds of newtons. The scaling is wrong. The numbers do not fit any intuitive model of what a small crustacean should be able to do. But here is where the story becomes genuinely extraordinary. The impact is only the first event. A fraction of a millisecond after the club connects, something else happens. A cavitation bubble collapses. Cavitation. The word sounds technical, but the phenomenon is both simple and spectacular. When any object moves through water at sufficiently high velocity, the pressure in the water immediately around that object drops dramatically. If the pressure drops low enough, the water locally stops being a liquid. It vaporizes. A bubble forms, filled with water vapor and dissolved gases, appearing almost instantaneously in the wake of the moving club. The bubble is under enormous stress. The surrounding water, still at ambient pressure, crushes in from all sides. The bubble collapses, and when it does, it releases energy. Not gently, not gradually. The collapse of a cavitation bubble is a violent event. It generates a secondary pressure wave, a shock wave, that radiates outward from the point of collapse. This shock wave can be just as powerful as the original physical impact of the club. It can produce temperatures approaching those found at the surface of the sun, though localized to a microscopic volume and lasting for an immeasurably brief time. It produces a flash of light, a phenomenon called sonoluminescence, visible only with sensitive instruments, and it can damage the hard shell of a crab or a clam, even if the club itself does not make direct contact. The prey is hit twice, once by the club and once by the physics. A snail closes its shell. It retreats inside its spiral fortress, confident in the millions of years of evolutionary pressure that have made that shell thick and hard and resistant. The mantis shrimp does not pry the shell open. It does not look for the soft opening, does not probe for weakness. It simply strikes the shell directly, and the combination of direct impact and cavitation shockwave shatters it. The snail did not evolve to survive an encounter with a shockwave. The dactyl club, the business end of this system, has been studied in extraordinary detail. At the microscopic level, it is a masterwork of natural engineering. The outermost surface, the impact region, is made of hydroxyapatite, a calcium-based mineral also found in bone and teeth, organized into a structure that is essentially a highly ordered crystal. Beneath this impact surface lies a region of layered fibers, helicoidal in arrangement, meaning they spiral around an axis like a twisted stack. These fibers are made of chitin, the same tough polysaccharide that forms the outer shell of most crustaceans, but arranged in a way that gives the material extraordinary resistance to fracture. The helicoidal arrangement means that when a crack forms, it does not propagate straight through the material in the direction of least resistance. Instead, it is redirected, forced to twist and spiral, requiring far more energy to advance. The crack is effectively confused by the architecture. This is not an accident. This is tens of millions of years of refinement, producing a material that can absorb enormous impact stresses without catastrophically failing. Engineering researchers have been studying this structure for years, not out of abstract curiosity, but because the architecture of the Mantis Shrimp Club offers direct lessons for the design of impact-resistant materials, for body armor, for aerospace applications, for protective structures of all kinds. The helicoidal fiber arrangement has now been deliberately replicated in composite materials, and the results have confirmed what biology worked out long ago. The geometry is exceptionally effective. But even the most sophisticated weapon is useless without a mechanism to deploy it. And the mechanism the mantis shrimp uses to generate its strike is no less remarkable than the club itself. Consider the problem. The muscles of a small crustacean are simply not powerful enough, contracting at the speeds that muscle tissue can contract, to accelerate the club to the velocities observed. If the strike depended on direct muscle contraction alone, it would be a fraction of the speed it actually achieves. The mathematics are clear on this point. Muscle power is not the limiting factor. Something else is happening. What is happening is a spring mechanism, a biological spring of exceptional sophistication. Inside the raptorial limb of the mantis shrimp is a structure called the saddle, a curved piece of mineralized material shaped somewhat like a hyperbolic paraboloid, the same geometric form as a riding saddle or a Pringle chip. This structure stores elastic potential energy. When the mantis shrimp prepares to strike, it contracts its muscles slowly, and these contractions compress the saddle and associated structures, loading them like a compressed spring. A latch mechanism holds the limb in the cocked position. The muscles contract, the energy is stored, and nothing moves. And then the latch releases. In an instant, all that stored elastic energy is transferred to the club, which accelerates with a force and speed that no muscle could produce directly. The system is a power amplifier. The muscles do the work over a relatively long time, the spring stores the energy, and then the latch releases it all at once, compressing the time over which that energy is delivered to nearly zero. This is the principle behind all spring-loaded systems, from a crossbow to a mousetrap. But in the mantis shrimp, it operates at a scale and speed and efficiency that no human engineer has yet matched in a mechanism of comparable size. The saddle itself has been analyzed in detail. It flexes when loaded and returns to its original shape when the energy is released, like a rubber band snapping back, but with far more precision and far less loss to heat. The material properties of the saddle are tuned to the task. It must be stiff enough to store energy efficiently, but flexible enough not to shatter under the stress of repeated loading. The mantis shrimp may strike many times in a single day. The spring mechanism must work reliably every single time. The entire limb is also constrained by geometry. The raptorial appendage folds back against the body, like the blade of a pocket knife, and when the latch releases, it unfolds along a precisely controlled arc. The path of the club is not random. It follows a trajectory determined by the anatomy, always arriving at the target along the optimal angle for impact. The mantis shrimp is not aiming in the way a predator with a brain-directed limb might aim. The anatomy does the aiming. The geometry of the joint and the shape of the appendage ensure that the strike arrives where it needs to arrive with high consistency. All of this happens in an animal that spends most of its life in a burrow. The burrow is worth considering on its own terms. Different species construct very different home environments. Some species, particularly the spearers, excavate burrows in soft sediment, digging out C-shaped or U-shaped tunnels that can be quite deep, with multiple entrances and a distinct internal structure. They maintain these burrows with considerable effort, regularly clearing debris and reinforcing the walls. Other species, the smashers in particular, often occupy crevices in the reef itself, sometimes enlarging existing holes, sometimes chipping away at the coral matrix to extend their space. The burrow is the center of the mantis shrimp's world. It eats there, sleeps there, mates there, and defends it with a ferocity that seems out of all proportion to the stakes involved. An intruding mantis shrimp, or almost any other animal that ventures too close to the entrance, will be met with a display of threat posturing. The animal will emerge from the burrow, spread the colorful fan of its tail, and often display the raptorial appendages in a way that makes the club clearly visible. This is not empty theater. The club is real, and the animal will use it. Animals much larger than a mantis shrimp have retreated from these displays. If the display does not work, the mantis shrimp will strike. And because mantis shrimp strike rivals during territorial disputes, as well as during prey capture, the durability of the club becomes relevant in a new way. The club has to survive striking not just the shell of a snail, but also the dactyl club of another mantis shrimp. These territorial fights are essentially hammering contests, with both animals striking repeatedly at each other's tail segments, which are the hardest and most heavily armored parts of the body. The tail in some species has been further reinforced with additional mineralization that seems specifically adapted to serve as a shield during these intraspecific combat situations. The visual system of the mantis shrimp has received almost as much scientific attention as the club, and for good reason. It is unlike any other visual system on the planet. Human eyes have 3 types of color photoreceptors, called cone cells, which are sensitive to different ranges of wavelengths corresponding roughly to red, green, and blue light. From these 3 receptor types, we construct our entire experience of color. We can distinguish somewhere in the range of a few million different colors, which seems impressive, until you learn that mantis shrimp have 16 types of photoreceptor cells. 16, and they extend the range of wavelengths those receptors cover far beyond what human eyes can detect, into the ultraviolet at one end of the spectrum and into deep red at the other. The initial assumption when researchers discovered this was that mantis shrimp must see a world of almost unimaginable color richness, that the 16 receptor types would allow them to discriminate colors with an ability that would dwarf any other animal. But the picture that emerged from behavioral experiments was more surprising and, in some ways, more interesting than that. When mantis shrimp were tested on their ability to distinguish between colors that are close together in wavelength, similar shades of the same color, they performed much worse than humans, much worse than bees, much worse than many animals with far fewer photoreceptor types. This seemed paradoxical, even contradictory. How could an animal with 16 photoreceptor types be less capable of fine color discrimination than an animal with 3? The answer appears to be that the mantis shrimp visual system is not optimized for fine discrimination. It is optimized for fast, efficient color identification. Rather than comparing the signals from different receptors to determine exact color through complex neural processing, the mantis shrimp appears to use its photoreceptors more like a color barcode reader, each receptor tuned to a specific wavelength range, and the pattern of activation across all 16 types producing an immediate and unambiguous identification of the color being viewed. This system trades precision for speed. In an environment where fast recognition is more valuable than nuanced distinction, where the difference between a prey animal and a predator must be resolved in milliseconds, having a system that produces an immediate answer may be more useful than having a system that produces an accurate answer after a moment of computation. The mantis shrimp lives in a world where moments of computation are precisely what it cannot afford. The ultraviolet sensitivity is particularly interesting in the context of communication. Many mantis shrimp species have ultraviolet reflective markings on their bodies, patterns that are invisible to most other animals on the reef, but clearly visible to other mantis shrimp. These patterns are used in communication between individuals, particularly in the context of territorial disputes and mating. They constitute a private visual channel, a language spoken between mantis shrimp that the rest of the reef community simply cannot read. Some species also possess circular polarization vision, the ability to detect whether light is rotating clockwise or counterclockwise as it travels. This is an extraordinarily rare visual capability. Most optical instruments cannot detect circular polarization without specialized filters. The mantis shrimp detects it naturally, with specialized regions of its compound eye, and mantis shrimp produce circularly polarized light from markings on their bodies, creating yet another private channel of communication that is invisible to everything else on the reef. The combination of 16 color receptor types, ultraviolet sensitivity, and circular polarization detection makes the mantis shrimp's visual world categorically different from that of any other animal we know of. We can describe its visual system in physical and biological terms, but we cannot meaningfully imagine what the mantis shrimp sees when it looks at a coral reef. The richness of information available to it from the light environment alone is simply beyond our cognitive reach. The life history of the mantis shrimp is rich with behaviors that reward close study. Mating is one area where the animal's complexity becomes particularly apparent. In many species, the mantis shrimp forms monogamous pair bonds of remarkable durability. A pair may stay together and share a burrow for 20 years or more. This is unusual in the invertebrate world, where most animals invest little in ongoing partnership. The mantis shrimp pair cooperates in the defense of the burrow, in the care of eggs, and in general maintenance of their shared territory. The bond between partners appears to involve recognition through both chemical and visual signals, and established pairs show coordinated behavior that suggests genuine familiarity with each other's presence and habits. When a female is ready to mate, the pair engages in elaborate courtship displays. The vivid coloration of the body and the tail fan are fully deployed. The raptorial appendages may be displayed in ways that emphasize their size and condition, which may serve as honest signals of the male's physical quality. After mating, depending on the species, either the female broods the eggs herself, carrying them in a clump held beneath her body and cleaned and aerated continuously, or the eggs are split between both parents, each caring for a clutch. Parental care in invertebrates is not common. In the mantis shrimp, it is practiced with a dedication that would not seem out of place in a far more complex animal. The juveniles, when they hatch, pass through a larval stage during which they are planktonic, drifting in the water column. During this period, they are prey for almost everything. The larvae are transparent, relatively helpless, and entirely dependent on the currents of the ocean for dispersal. Many will be eaten, many will drift to habitats unsuitable for settlement. Only a fraction will survive to find suitable substrate, settle, and begin the process of growing into the formidable adults they have the potential to become. The growth process itself is interesting. Like all crustaceans, mantis shrimp must molt, shedding their exoskeleton to grow. The molting process is dangerous. For the period immediately after a molt, before the new shell has hardened, the animal is soft and vulnerable. The club is temporarily useless. The armor is gone. An animal that has evolved into one of the reef's most aggressive and capable predators becomes, for a brief and humbling window of time, genuinely defenseless. Mantis shrimp typically remain deep in their burrows during this period, sealing the entrance if possible, and waiting for the new shell to harden before venturing out again. The chemistry of shell hardening in mantis shrimp is an area of active research, not only because of interest in the animals themselves, but because the process by which they deposit mineralized material in such precisely organized structures has implications for understanding how biological systems control mineralization more generally. The mantis shrimp does not simply calcify its exoskeleton uniformly. Different regions of the body receive different amounts and different types of mineralization, with the dactyl club receiving the most intensive treatment of all. Understanding how the animal controls this process, what molecular signals determine where and how much mineral is deposited, is a question that connects to fundamental problems in materials science and biology alike. On the reef itself, the mantis shrimp occupies an ecological position that is easy to underestimate. Because they are small and secretive, spending most of their time inside burrows, they are easy to overlook. A diver swimming over a reef might never see one. But they are common. On healthy tropical reefs, mantis shrimp can be among the most abundant predators, present in densities that mean they collectively consume an enormous amount of prey. They are important regulators of populations of snails, crabs, clams, and small fish. Removing them from a reef community, or allowing their populations to decline, would have consequences that ripple through the food web in ways that might not be immediately obvious, but that would eventually be felt by everything connected to the species they normally keep in check. They are also prey themselves. Octopuses, large fish, and some species of predatory shrimp will all eat mantis shrimp, given the opportunity. The mantis shrimp's first line of defense is its burrow. Its second line of defense is its awareness. The eyes that can detect ultraviolet light and circular polarization are also highly sensitive to movement and to changes in light intensity, giving the animal early warning of approaching threats. When an octopus reaches into a burrow entrance, it may encounter a strike from the mantis shrimp within, a strike that can draw blood and cause genuine damage to the octopus's arm. Many predators learn quickly that burrows containing mantis shrimp are better left alone. The relationship between the mantis shrimp and the reef around it is also mediated by the burrow in a physical sense. In soft sediment habitats, the excavation of burrows by mantis shrimp creates channels for water movement and oxygen exchange within the sediment. This bioturbation, the disturbance of the substrate by animal activity, has ecological consequences for the community of animals and microorganisms living in and on the sediment. The mantis shrimp is not only a predator, but an ecosystem engineer, reshaping its immediate environment in ways that affect the species that live alongside it. In coral reef ecosystems, where the layered complexity of species interactions creates a web of interdependencies that scientists have only partially mapped, the mantis shrimp threads through multiple levels of that web simultaneously. It eats, it is eaten, it burrows, it fights, it mates and broods and cleans and defends. Each of these activities connects it to other species, other processes, other dynamics. It is a small animal in a vast and complicated system, but it is not an incidental one. There is a particular species that has become, in popular science communication, the representative of the entire group. Odonodactylus ciliaris, commonly called the peacock mantis shrimp, is a smasher species that grows to around 15 to 17 centimeters in length and is covered in some of the most extravagant coloration found anywhere in the ocean. The base color of the body can be a deep emerald green. The legs are bright orange or red. The raptorial appendages are edged in electric blue, the tail fan carries concentric rings of vivid color, and the compound eyes are large and protuberant and move independently of each other, scanning the environment with an unnerving thoroughness that gives the animal an almost alien quality when you observe it at close range. The peacock mantis shrimp inhabits tropical reefs across the Indo-Pacific, from the Red Sea and East Africa across to Hawaii. It is found in shallow water, typically at depths of less than 40 meters, and often in areas of mixed coral, rubble, and sand, where suitable burrow sites are available. It is a moderately large animal by mantis shrimp standards, but not the largest species known. That distinction belongs to species in the genus Lisiosquilina, the spearers, which can reach close to 40 centimeters. The peacock mantis shrimp has been the subject of numerous research programs precisely because it is large enough to be worked with experimentally and vivid enough to attract interest. Studies on strike force, on club material properties, on visual physiology, and on behavior have all used this species extensively. In captivity, it is known for its territoriality and its willingness to strike at the acrylic walls of aquarium tanks, which has occasionally resulted in cracked tanks and genuinely startled aquarists. Experienced keepers know to house mantis shrimp only in the most robust enclosures and to handle them, if handling is necessary at all, with tools rather than unprotected hands. The strike force measurements that have attracted so much attention were largely derived from this species. When researchers positioned force transducers in enclosures occupied by peacock mantis shrimp and induced the animals to strike them, the results consistently demonstrated forces far beyond what the animal's mass and musculature would predict through naive scaling arguments. The peak forces measured in the most rigorous studies have been reported at close to 1,500 newtons in larger individuals, though there is variation, and the exact numbers depend significantly on the methodology used. Even the lower end of the measured range, several hundred newtons, would be astonishing from an animal of this size. What does a force of that magnitude mean in practical terms? A moderate-sized hermit crab, an animal that has presumably evolved some resistance to the kind of predation pressure that mantis shrimp represent, can have its shell completely shattered by a single well-placed strike. A clam that takes considerable effort to pry open with a specialized tool can be cracked open in a fraction of a second. A human finger, positioned incautiously near a striking mantis shrimp, can be split open to the bone by an animal that the person may not have even properly seen before the strike landed. These are not exaggerations produced for effect. These are documented outcomes from real encounters between humans and mantis shrimp, most of them in the context of marine biology fieldwork or the transportation of live specimens. The cavitation aspect of the strike adds a dimension that makes the raw force measurement somewhat incomplete as a description of what the animal can do. Because the cavitation bubble's collapse produces a secondary pressure event that arrives at the target surface independently of the direct impact, the effective damaging power of a strike is greater than what the force measurement alone captures. The target is subjected to a pressure pulse sequence, first the club, then the shock wave, arriving in such rapid succession that for practical purposes they are simultaneous but physically distinct. Materials that might absorb or deflect a single impact can still be damaged by the combination. This has been observed directly in laboratory settings. Researchers have placed targets at distances from a striking mantis shrimp at which the club itself does not physically contact the target, but the cavitation event still occurs nearby. The shock wave from the bubble collapse can cause measurable damage to biological material at these distances. The effective kill zone of a mantis shrimp strike extends slightly beyond the physical reach of the club itself. In the ocean, this matters. A small fish hovering a fraction of a centimeter too close to a burrow entrance may not need to be struck to be stunned. The shock wave may be enough to disorient it, to disrupt the coordinated muscle action that keeps it hovering in the water column, to buy the mantis shrimp the tiny additional moment it needs to close the gap and make the follow-up strike. Predation in the ocean operates on margins so thin that any advantage, even a marginal extension of effective strike range, translates directly into more meals and greater survival. It is worth pausing to consider the energetics of what the mantis shrimp does. Every time it cocks and fires its spring mechanism, it expends energy. The muscles that load the spring, that compress the saddle and related elastic structures, must contract. Muscle contraction requires metabolic energy. ATP, the cellular currency of biological work, is consumed. The animal must eat enough to support not only its basic metabolic needs, but also the cost of repeated striking. For a predator that feeds on hard-shelled prey, which may require multiple strikes to overcome, the energy budget is not trivial. This is one reason why the spring mechanism is so important beyond simply enabling higher strike velocities. By allowing the muscles to do their work slowly, the spring mechanism means the muscles are operating in a regime where they are maximally efficient, converting chemical energy to mechanical work with minimal waste. If the muscles had to power the strike directly, contracting rapidly in the time available, they would be working far from their optimal mechanical conditions, and the efficiency would be much lower. The spring is not just a speed amplifier, it is also an energy efficiency device. The material science of the club are still being actively investigated, and each new analytical technique that becomes available tends to reveal another layer of structural sophistication. Synchrotron X-ray diffraction has revealed the orientation of crystals within the impact region at micrometer resolution. Nano-indentation testing has mapped the mechanical properties of different regions of the club at scales approaching individual crystal grains. Electron microscopy has revealed the precise geometry of the fiber helix in the underlying layer. What has emerged from all of these studies is a picture of a material that is not simply strong, not simply tough, but optimized in a way that addresses multiple conflicting demands simultaneously. A material that is maximally hard is typically brittle. A material that is maximally tough, meaning it absorbs energy rather than transmitting it, is typically soft. The Mantis Shrimp Club is hard at the surface to maximize the transfer of impact energy to the target without deformation, and progressively more energy absorbing in the interior layers to prevent cracks from propagating through the full thickness of the club. It manages the inherent trade-off between hardness and toughness by varying both properties spatially across the cross-section of the club, placing hard material where it is needed and tough material where it is needed. This principle of spatially graded mechanical properties has become a major theme in modern materials engineering. Composite materials designed with property gradients, rather than uniform compositions, have shown superior performance in impact resistance, in ballistic protection, in the tolerance of thermal stress, and in resistance to fatigue under cyclic loading. The Mantis Shrimp Club did not inspire these engineering developments directly, because engineers were pursuing gradients in parallel with the biological research. But the Mantis Shrimp has served as a validation and an inspiration, demonstrating that the concept is not merely theoretically sound, but biologically proven over geological timescales in a context of extreme mechanical demand. There is also the question of what happens to the club over time. A structure that is used repeatedly to strike hard surfaces, generating forces of hundreds to thousands of newtons, is subject to cumulative damage. Metals fatigue under cyclic loading. Ceramics accumulate microcracks. Polymers creep. What happens to the Mantis Shrimp Club? The answer involves the molting cycle. Because mantis shrimp regularly shed and regrow their exoskeleton, any accumulated damage in the club is periodically renewed. The animal does not need to maintain the club in perfect condition indefinitely. It needs to maintain it through a molting interval, which might be weeks to months, and then it gets a fresh start. The molt cycle functions as a reset mechanism for the material integrity of the club, a biological solution to the fatigue problem that simply does not apply to engineering structures, which do not molt. This has interesting implications for how we interpret the material properties of the club. The optimization may not be for indefinite durability, but for performance over a single molt cycle, combined with repairability through the periodic renewal that molting provides. This is a different design philosophy from what engineers typically apply, where structures are generally expected to function indefinitely, or at least over timescales of years or decades without replacement. The Mantis Shrimp operates on a subscription model, rebuilding its tools regularly, rather than trying to build a tool that never wears out. Looking at the broader evolutionary context of the Mantis Shrimp's weapons and abilities helps situate what is remarkable about them. Natural selection does not design. It selects individuals that are better at catching prey, better at defending their burrow, better at surviving encounters with predators, contribute more offspring to the next generation. Over millions of generations, the average member of the population tends to improve in these capacities. Adaptations that improve performance in critical behaviors are reinforced. Adaptations that reduce performance are eliminated. The mantis shrimp has been under selection pressure for approximately 400 million years. 400 million years of individuals competing for food, for burrows, for mates. 400 million years of the slightly faster striker eating more, and the slightly weaker club breaking at the wrong moment. The result is not a design produced by an engineer working from first principles, but an outcome of a process that is in some ways more demanding than engineering, because the stakes are absolute and the testing is continuous and merciless. Every mantis shrimp alive today is descended from an unbroken chain of successful predators and successful breeders, stretching back to before the Devonian period. Every link in that chain was good enough to survive and reproduce. The accumulation of small advantages over 400 million years of selection has produced the striking mechanism, the club material, the spring, the latch, the eyes, the colors, the territorial ferocity, and all the other features that make this animal so remarkable. No single generation of engineers working on a single project could produce an outcome refined by that depth of time, and yet engineers have learned from it, continue to learn from it. The mantis shrimp is an existence proof, a demonstration that certain solutions to certain engineering problems are achievable, that the physics and materials science of high-velocity impact in small structures can be managed, that lightweight, high strength, excellent toughness, and power amplification through spring mechanisms can all be combined in a package that fits in the palm of a hand. Body armor inspired by the helicoidal fiber architecture of the Mantis Shrimp Club is under development in multiple research groups. Materials for protective helmets, for crash structures in vehicles, for the housing of sensitive electronic equipment that must survive impacts, have all been influenced by the biological template. The saddle spring mechanism has been studied as a model for microsystems where high-force, short-duration actuation is needed, applications in micro-robotics, and in minimally invasive surgical tools where large forces must be delivered through tiny structures in very short times. None of this research would have been possible without the mantis shrimp. More precisely, none of it would have happened at the pace it has without the convergence of biological curiosity and engineering ambition around an animal that is small enough to overlook and extraordinary enough to demand attention from anyone who takes the trouble to look. There is something instructive about the mantis shrimp as a symbol of what the natural world contains. It lives in crevices and reefs that are themselves under threat, in oceans that are warming and acidifying, in ecosystems that are being altered faster than science can document the changes. The loss of coral reefs, which the human-driven carbon experiment is driving with brutal efficiency, means the loss of habitat for the mantis shrimp and for the thousands of other specialized animals that depend on reef structure. An animal that has survived 400 million years of geological upheaval and mass extinction events is now contending with a trajectory of change that is geologically instantaneous. The reef where the mantis shrimp strikes its prey, where it defends its burrow, where it sees colors and patterns invisible to every other eye on the planet, is not an abstraction. It is a specific and irreplaceable ecosystem produced by millions of years of biological accumulation, housing thousands of species in an interdependency so complex that disrupting any part of it sends consequences propagating outward in ways that cannot fully be predicted. The mantis shrimp is part of that system. It is connected to everything around it through the web of eating and being eaten and competing and cooperating and modifying the substrate and providing nutrients through its waste and its body when it dies. To understand the mantis shrimp is to begin to understand the reef, and to understand the reef is to begin to understand what is at stake when that reef is damaged, bleached, dissolved, smothered in sediment, dynamited for fish, or harvested past the point of recovery. The physics of the mantis shrimp strike are a window into the reef, and through the reef into the ocean, and through the ocean into the systems that maintain the conditions under which life on this planet is possible at all. Standing back from the mathematics and the material science and the evolutionary history, what is left is something simpler and perhaps more important. An animal the size of a human hand, brightly colored in ways that seem almost deliberately excessive, hiding in a crack in the reef, possessing eyes that perceive a universe of color no human will ever experience, capable of delivering a blow that generates a shock wave. That shrimp has been doing this, in one form or another, for longer than there have been trees on land. For longer than the Atlantic Ocean has existed, for longer than the Rocky Mountains have been mountains. It is here now, in the shallow, warm water, in its burrow, cleaning its eggs, or watching the water above the entrance with those extraordinary compound eyes, waiting for the next snail to pass within range. When it does, the saddle will compress, The latch will hold for a fraction of a second while the muscle loads the spring, and then the latch will release, and the club will travel 20 meters per second toward the shell, and the cavitation bubble will form and collapse, and the shell will crack, and the mantis shrimp will drag the animal inside its burrow and eat. It will do this today, and tomorrow, and every day until it molts, and then it will rebuild its club and do it again. It will do it for years, or perhaps decades, paired with the same partner, in the same burrow, on the same reef, conducting a life of extraordinary precision and capability in a space measured in centimeters. The ocean is full of such creatures, animals that are easy to miss and impossible to fully comprehend once you have found them. The mantis shrimp is simply one of the most honest examples. It does not hide what it is. It is vivid and aggressive and physically improbable and deeply strange. It invites exactly the response that the best of the natural world always invites. The response that makes you stop whatever you are doing and simply try to take in the fact that this animal is real, that it exists, that it punches with a shockwave, and that the reef it lives in is worth understanding, worth studying, and worth keeping.